It gives me great pleasure to introduce Dr. David Lynch, who's a neurologist who works at the National Hospital for Neurology and Neurosurgery. He also leads the adult IWMD service for adults, and he's going to talk to us through clinical trials. Yeah, or you can use the 101.
There we go. Hi everyone, so thank you so much for asking me to speak. My name is David Lynch. I'm a neurologist at Queen's Square in London, and I lead the Inherited White Matter Disease Service with many colleagues who are here today. So Karen asked me to speak about clinical trials, a sort of basic explanation for patients and families. I know that many families who Alex TLC support actually do loads of research themselves and sometimes know a lot more about the disease than individual clinicians or doctors. So apologies if it's a little bit basic for some people, but I wanted to try and make sure it was accessible to the general audience. So clinical trials are really important. We really can't drive science and medicine forward without them, but we need participants to take part in clinical trials for them to take place. So understanding and knowing how clinical trials work is important for patients and families so that they can decide if participating in one is right for them. So what a clinical trial is research study involving people in which you're testing some kind of intervention. And we typically think of them as being medications, but you can do a clinical trial for a medical device, something that you implant, a surgical procedure, or even talking therapies like psychological therapies. But clinical trials are really important to make sure that the treatments that we use work and that they're safe, and that they help us to find ways to treat or diagnose or prevent diseases and to drive medicine forward. So in a clinical trial, there's one sort of central question that everything else comes secondary to, and that central question is sorting out effective from ineffective treatments. So we want to show that an effective drug works, or that an ineffective drug does not work, and that we're not going to go forward and use it again. And when you design a clinical trial, you really want to avoid two things. One is getting rid of a truly effective drug just by accident, or approving a drug that actually doesn't work and wasting everybody's time taking it and then it being withdrawn from the market later. So clinical trials are really designed around safety first and then efficacy second, and they're divided into phases, and at the end of each phase the researchers will take a deep look at the information that's come out and make sure that it's safe to go on to the next phase and give it to more people. So it's divided into phase 1, which is all about safety. I'll talk a little bit more about these. Phase 2, which is also starting to be a little bit more about efficacy of the drug, but also side effects. Phase 3, sorry, this is on auto advance, that's just changing it. Phase 3, which is more about comparing drugs and making sure that they work, or maybe comparing your new drug to an existing drug. And then in phase 4, this is when the drug is actually on the market and people are using it and prescribing it. It's been given to lots more people over a longer period of time, and so you're getting real-world information. People often ask about placebos and why there would be a placebo arm in a trial, or they feel like they want to be in a trial but they really don't want the placebo, or why can't I get the drug and I'm not the placebo? The default position generally is that trials would have a placebo control to compare against your drug, or to the placebo arm might actually be the current best available treatment. And the reason that you need to have a placebo generally is because sometimes people just feel like they're getting better just because they're interacting more with healthcare. So if you're coming up every month for an infusion of a drug and you're seeing a study nurse or a doctor every month, you might feel like you're doing better than if you just have a annual appointment with your doctor. So for that reason, sometimes people in clinical trials can feel better even when the drug is not doing anything. So it's important to have a placebo. But sometimes it's not possible to have a placebo, and this applies particularly for rare diseases like leukodystrophies often, because you just might not have enough people to put into the trial so that you can have two groups. So you might just have to have a single group of everyone getting the treatment, and I'll talk a bit more about that later. So if you do have a placebo, then it's important to randomly divide people to whether they receive the treatment or the placebo. You really want the two groups to be as similar to each other as possible. So you don't want one group where people all have mild symptoms and the other group people who have severe symptoms, because those two groups can't be compared to each other very well. So you want the two groups to be similar in sort of their age, how severe the symptoms are, how long they've lived with the illness. And so the best way to do that is just to put all the patients together in one group and then just assign them completely at random so that you try to minimize the differences between the two groups. So in a phase 1 trial, this is all about safety. And usually this is the first time you're giving the drug to a human being. So you're going to generally give it to healthy people, just completely healthy volunteers. And you look at lots of different effects. So what the drug is doing to the heart, to the kidneys, to the liver, whether it's causing any side effects or that makes people feel sick or dizzy, things like this. You're also monitoring how the drug is used by the body, how it's broken down, how long it lasts for in the body, these kind of things. So it's important to do the phase 1 in healthy people generally before you're going to move on and give the drug to people who are living with a condition that might be a serious condition affecting their health. And once you get through phase 1, it looks like the drug is safe to give to people. You can then go on to phase 2, which again is usually more about side effects as well, but you might start to see some evidence of whether the drug works, so some efficacy. So you're generally giving it to more people, but the number of people that will be in the trial will completely depend on the condition you're treating. You know, if you're treating a new blood pressure drug, you might give it to hundreds of people, but if you have a leukodystrophy, it might be more like 10 people or 20 people if it's a very rare condition. So sometimes in a phase 2 trial, also what you'll look at are things called biomarkers, which are things you can measure maybe in blood or in spinal fluid or an MRI scan that can give you a sense of whether the drug is having an effect before it would be noticeable by the patient. You know, sometimes it takes longer for you to notice change in symptoms, maybe more like 6 months, a year, 2 years, but you might see a change in a biomarker earlier. So in a phase 2, you're also generally considering monitoring for more side effects, because you might start to get side effects in patients who have the condition that you didn't see in healthy controls or healthy volunteers. And then once you get through phase 2, you can move into the phase 3 part of the trial, which is usually much bigger. And the phase 3 trial is usually considered important to try and get the drug approved. Again, I say test in large groups, but it will depend on how many people you can get into the trial. And sometimes you might be comparing whether your treatment is better than what's available rather than to a placebo. And because you have more people in the trial, you now will see more rare side effects. And at the end of the phase 3, if it looks like the drug is working, then it will likely be approved, and you can go on the market and it can be prescribed more widely and used in more people. And then we enter the phase 4, which is post-marketing monitoring. So this is where you're continuing to gather information on the drug. You're also going to get your first information maybe on how the drug should be used in the real world, because a clinical trial is in some ways a kind of quite synthetic or idealized way of providing care. So things are messier in the real world, and you might start to notice things about the drug that you didn't see in the trial. So we do continue always when the drug is on the market to monitor it, and if it looks like there are problems arising, then we can deal with them. It might be changing how the drug is used, limiting it. Sometimes it might be taken off the market if problems come up later. Sometimes people ask, there's a clinical trial going on, but I'm not able to join it, and why is that? So I wanted to talk a little bit about why you focus on certain patient groups, and that's called eligibility. So usually when you're designing a trial, you need to catch patients in the right phase of the disease. So usually we're focusing on patients who are in the early phase or the moderate stage of the disease, rather than patients who have a very, very advanced disease. And the reason for that is that it's generally easier to measure change in someone who has mild symptoms or moderate symptoms. If someone has very severe symptoms, then it can be very difficult for us to see whether anything is changing in that. So as well as trying to get people at the right sort of phase of the disease early on, you also want to try and make the patients as similar to each other as you can, because that reduces the variation. So you're trying to get patients all kind of with similar level of symptom severity, who've maybe had the disease for the same length of time, and not patients where there's one patient who's very, very severely affected and one who's very mildly affected, because then you just have this huge variation, and then it can make it very foggy and difficult to see whether the drug is having an effect. So by doing this, we design what we call inclusion and exclusion criteria. So the inclusion criteria, what gets you included into the trial. Exclusion criteria, things that you might have that will mean that you're not a good candidate for the trial. So normally, you're going to not want to put patients into a trial who might be very medically unwell for some other unrelated reason. So if someone has a leukodystrophy, but then they have a separate condition as well that might make them quite unwell and might make them maybe be at more risk from the trial. So as well as protecting the trial, it's also about protecting the patients. You want people to be generally stable before they enter a clinical trial where they're getting a new drug. So to design the inclusion and the exclusion criteria, the sponsor who's running the trial, which is usually a company, but it could be a university group, they will meet with experts and will sit down and will try and decide who should this drug be tried in, what are we going to measure, because there's endless things you can measure about people, whether it be things you can measure clinically about them, or things you can scan, or blood tests you can do. So you want to know what you're going to measure, and then what change in those measurements is going to mean something. So how will we know whether the drug is effective? And usually when the trial is being designed, there can be input from regulators who decide by themselves independently at the end whether the drug gets licensed. They might have some things they want the company to include to show them or prove to them that we think the drug works. There are some important differences with gene therapy trials. So we know that leukodystrophies are genetic, of course, caused by DNA faults. And so gene therapy is a very exciting way to try and repair DNA, or kind of really get to the basic level of what's happening in the condition. And gene therapy is very, very exciting, but it's an area that we have to be quite cautious in. And so gene therapy has some particular differences to trials of more common things like, for example, blood pressure medications. And that's because gene therapy has the potential to cause a change that is permanent and irreversible. And so in a gene therapy trial, you're generally not going to give the drug to a healthy volunteer as you would in a normal phase 1 trial, because that could cause permanent changes to that healthy person. So in a gene therapy trial, often the phase 1 includes patients who have the condition. And they might be done together. You might have a phase 1 and phase 2 kind of combined in patients. And you need to manage specific risks with gene therapy trials. So as I said, it's a very exciting area, but it is one that we're very cautious of, because when you give a gene therapy, you can cause immune reactions in people, because you generally use a virus to give the genetic material into that person, and sometimes people have reactions to the viruses. You're at risk of causing mutagenesis, which is where you cause a change in DNA that you didn't expect, and that could be permanent and cause problems. And also you have unknown how long the gene therapy lasts for. So some gene therapies are kind of promoted as a cure, although we don't really like using that word, but they're thought to have a permanent effect, but we don't really know whether the effect might wear off in 10 years. So generally in gene therapy trials, you need to follow patients up for a long time, often 15 years, to measure the effect out. So moving on to natural history studies. So these are a different type of study where you're not giving an intervention or a treatment, but where you're just tracking how the disease changes over time. And often we will know that a condition might progress. We will see patients with the condition and we know, okay, it will maybe be worse in a year or in 2 years, but we don't know exactly how much worse, or we don't know how much the MRI will change. In a 12-month period, will the MRI change by 5% or 10% or 15%? So a natural history study is where you're just tracking people with the condition over time, not giving them a drug, and then just measuring important things about them. And sometimes a natural history study becomes very important as a comparison when you're running a trial that doesn't have a placebo. So you might have a very rare condition where it's very hard to get enough people into the trial to actually divide them in two and have one group get the treatment and one group get the placebo. So you run a trial where everybody gets the drug, and then you're comparing it to, say, information that you got from a natural history study. So that can be really, really important. I'd encourage people, if they can, if they have an opportunity, to take part in natural history studies, because the more of them we have, and the more information we have, the faster we can make progress. So that just brings me to near the end and the role of participants. We really wouldn't be able to run trials without patients and families. So we know people who run trials how burdensome it can be to take part in a trial. So it can take a lot of your time, it can take a lot of energy, it might make you very tired, it might be a lot of travel, might be a lot of tests when you come in for the assessments. But we try our best to make sure that it's as easy as possible to take part in a trial. We're very aware that you're giving your time and your energy to do this, and especially when you might be coping with a condition as well. So we try and minimize that burden if we can. So how you can get involved, if you'd like to, is to speak to your doctor, to your specialist. Is usually going to know what trials are going on, either locally or maybe even in other places or other countries. You can have a look online yourself. So there's a website called clinicaltrials.gov where companies and universities will register their trial and give information about it. So you can look up, and it will often say when it's open, or if it's closed, or if it's an old trial. You can, of course, talk to Alex TLC, because they know everything. And then you can try and understand yourself about what it might take to be in this trial. How many visits are there? How often do I need to be seen? How much time will it take? The clinical trials in leukodystrophies are slow, but we're making progress, and we have had some success stories, like with Libmeldy for metachromatic leukodystrophy, which is available. And we have some current trials at UCLH, just to mention. We have the IGNITE trial in CSF1R related ALSP. We have an Alexander disease, adult onset Alexander disease natural history study. I think you might hear more about that from my colleague Dr. Wade later. And of course, we're involved in the Ionis Alexander disease gene therapy trial as well. So just to bring to the end, clinical trials are really important to make advances in medicine, and they're focused primarily on the safety. So that's hopefully reassuring that this is our main priority. You're not going to be, at risk in a trial generally. You're not sort of a guinea pig, as people might say, but they're really designed carefully to be safe. And we do need to make sure that we get the right people into trials so that we get the most information out of them. So for you, the takeaway I think should be to try and stay informed about trials that are happening, stay up to date. And if there are trials happening, to think about joining them and talk to your doctors.